And welcome to my channel Sam Biology Class. Friends, today we will discuss about DNA library. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. DNA library or gene library is a collection of DNA fragments that are cloned into a population of identical vectors and are stored within suitable host organism. There are two types of DNA library. First one is genomic library and second one is cDNA or complementary DNA library. Today we will know more about genomic library. Genomic library is a collection of overlapping DNA fragments that are cloned into a population of identical vectors and are stored within suitable host organisms such as E. coli or yeast. Construction of genomic library involves several steps. First step is the extraction and purification of genomic DNA. Second step is the partial digestion of genomic DNA into fragments of similar size by using restriction endonucleases. Each of these fragments contain one or more genes. In third step, these DNA fragments are inserted into cloning vectors which were cut using the same restriction enzyme. During insertion, DNA ligase is used to seal the gap. This process, known as insertion or ligation, results in the formation of a pool of recombinant vectors, each of which contain a different DNA fragment. In last step, these recombinant vectors are introduced into E. coli cells through transformation, creating genomic library. Now let us discuss about the construction of genomic library in greater details. Construction of genomic library begins with the extraction of genomic DNA from the cells of the organism. Genome includes both coding and non-coding region. DNA can be extracted from cells by different methods such as cell lysis, protein digestion and phenylchloroform extraction. We know that genome consists of very large DNA molecules which cannot be inserted directly into the cloning vector. Because of this reason, the genomic DNA is cleaved into fragments of suitable size and then the DNA fragments are inserted into a cloning vector. DNA can be fragmented by different methods such as sonication, mechanical shearing or enzymatic digestion using restriction enzyme. In this example, the genome is subjected to partial digestion by using restriction enzyme. Restriction enzyme, also known as restriction endonuclease, cleave DNA at specific recognition sites. This partial digestion results in the formation of a number of DNA fragments. In the next step, these DNA fragments are inserted into a suitable cloning vector. Genome size varies among organisms, so cloning vectors must be selected accordingly. Different cloning vectors are available for the construction of genomic library. These vectors are plasmid, phage lambda, cosmid, bacteriophage P1 vector, P1 artificial chromosome, bacterial artificial chromosome and yeast artificial chromosome. In this example, plasmids are used as the cloning vector. What is plasmid? Plasmids are small extrachromosomal and circular double-stranded DNA molecules. There are two types of plasmids, natural plasmid and artificial plasmid. Natural plasmids exist in nature and are found in bacterial, archaeal and some eukaryotic cells where they confer antibiotic resistance and other advantages to the host. Artificial plasmids, on the other hand, are engineered in laboratory by combining DNA segments from different natural or artificial plasmids along with regulatory elements. Artificial plasmids are often used to clone small DNA fragments. In our example, these are the DNA fragments which are obtained from the partial digestion of genomic DNA. So all these fragments together make up the total genomic DNA of the organism. After fragmentation of genomic DNA and selection of the cloning vector, the next step is to insert the DNA fragments into the vector, which in this example is plasmid. Plasmids are circular double-stranded DNA molecules. In order to insert these DNA fragments into the vector, plasmids are first cleared by using suitable restriction enzyme. Same restriction enzyme should be used for fragmentation of genomic DNA and also for the linearization of the plasmids. Restriction enzyme recognize specific sites on these plasmids and cleave at that point, thereby yielding sticky or cohesive ends. As a result of cleavage at the specific recognition site, the plasmids now become open or linearized, after which the DNA fragments can be easily inserted into these linearized vectors. For insertion, these plasmids are mixed with DNA fragments and incubated under suitable conditions in the presence of enzyme DNA ligase. During incubation, these DNA fragments are inserted into the linearized plasmids and then DNA ligase seals the gap. This process, known as insertion or ligation, results in the formation of a pool of recombinant vectors. This is the pool of recombinant plasmids, each of which contains a different DNA fragment. Following this, these recombinant DNA molecules are introduced into a suitable host, such as E. coli or yeast. Recombinant vectors are introduced into competent E. coli cells through a process known as transformation. Transformation commonly uses methods such as heat shock or electroporation. Both these methods induce the formation of transient pores in the cell membrane through which recombinant vectors easily enter into the E. coli cells. This results in transformation. Transformation is not 100%. That means some of the E. coli cell fails to uptake the recombinant vector. As a result, the cell remain non-transformed. Remaining E. coli cells which have successfully incorporated the recombinant molecules become transformed. In the next step, we have to identify the E. coli cells which have taken up the recombinant DNA. For selection of transformed E. coli cells, this bacteria is grown on an agronutrient medium containing a specific antibiotic. In such a medium, only transformed bacterial cells which are resistant to antibiotic will be able to grow and will form colonies. Remaining bacterial cell which is non-transformed will die. All these colonies together make up the genomic library. Next step is amplification. The genomic library can be amplified by growing the surviving colonies in a large-scale culture. Once the colonies have been grown, the library can be screened for the presence of a specific DNA fragment or a gene which has been incorporated into the vector. Genomic library has several applications such as genome sequencing, identification of DNA, comparative genomics, and study of regulatory elements. Friends, this is all about Genomic Library. For more updates, subscribe my YouTube channel, Sam Biology Class.